Good day, everyone. I'm Sir Franz, and welcome to our lesson on lipolysis and ketogenesis. First, we have to remember that the form of stored fat is triacylglycerol. During a fast or a period where you don't eat, stored fat or triglycerides are hydrolyzed to release three fatty acids and a molecule of glycerol from the original form of the triglyceride. So the reaction sequence is simple. Triglyceride being broken down by hormone-sensitive lipase, producing a molecule of glycerol and three molecules of fatty acids. HSL or hormone-sensitive lipase is specific. It removes only fatty acids from carbon number 1 and carbon number 3 of the glycerol molecule. HSL is activated by glucagon and epinephrine, which are also known as counter-regulatory hormones, which we have discussed in our previous lesson. So, glucagon and epinephrine not only increase the sugars in the blood, but they also facilitate lipolysis or the breakdown of fat. This process is inhibited by insulin, and the additional lipases remove the remaining fatty acids. So let's now turn our attention here on the image on the right. We start with the high level of epinephrine, which occurs when you don't have something to eat. It binds on its cell surface receptor and then catalyzes the conversion of adenosine triphosphate into a molecule of CAMP, cyclic adenosine monophosphate. This cyclic adenosine monophosphate is actually the second messenger, which then activates protein kinase. This protein kinase will facilitate phosphorylation of hormone-sensitive lipase, which means protein kinase will cause the attachment of a phosphate group to hormone-sensitive lipase. Once hormone-sensitive lipase is phosphorylated, it is now turned on or activated. And that's the time it will catalyze the hydrolysis of triglycerides into glycerol and fatty acid. On the other hand, if you have insulin, insulin will cause an increase in the activity of phosphatases, which will remove phosphate through a process known as dephosphorylation. And if hormone-sensitive lipase is dephosphorylated, it does not have a phosphate group, it is turned off. And therefore, lipolysis is also turned off in the presence of insulin. So, for lipolysis to occur, you need to have low levels of insulin and high levels of the counter-regulatory hormones. The released fatty acid will then be catered by the mitochondrion through beta-oxidation. Beta-oxidation will remove acetyl coenzyme A from the ends of fatty acids, but at the same time, it will also produce reduced NAD and reduced FAD. The acetyl coenzyme A produced will enter the Krebs cycle, and the reduced NAD and reduced FAD will then enter the respiratory chain to produce ATP. Beta oxidation occurs in tissues that utilize fatty acids as energy source, primarily muscle cells and liver cells. Fatty acid activation occurs in the cytosol, whereas its consequent beta oxidation, or the actual reaction process, occurs in the mitochondrion. We start with palmitic acid as the substrate. Remember that palmitic acid is a long-chain fatty acid. It's 16 carbons long, and it's the primary fatty acid that we see in triglycerides. And we end up with 8 molecules of acetyl coenzyme A, which is 2 carbons long, 7 molecules of reduced NAD, and 7 molecules of reduced FAD, which means for a 16-carbon fatty acid, we perform 7 cycles of beta oxidation. The rate-limiting step, which is the slowest step, would be to actually transfer the fatty acyl coenzyme A from the cytosol where it is activated into the mitochondrion. So it's the transfer which is the slowest step. And the enzyme that catalyzes this would be CPT, carnitine palmitoyl transferase. So let's now go into the important steps of beta oxidation. The first one would be to transfer the fatty acyl-CoA from the cytosol into the mitochondrial matrix. And remember, this is the slowest step. So we first orient ourselves. On the left, it's the cytosol, the liquid portion of the cytoplasm. And then you have here to encounter the outer mitochondrial membrane. So this is already the walls of the mitochondria. The outer mitochondrial membrane going to the right, and then the intermembranous space in the middle, the inner mitochondrial membrane, and then the mitochondrial matrix. So our goal here would be to transfer the fatty acid from the cytosol into the mitochondrial matrix. First, we need to activate the fatty acid. We need to give it energy. And to do that, we hydrolyze a molecule of ATP and we form a bond with coenzyme A to produce fatty acyl coenzyme A. The energy from ATP is contained between the bonds of the fatty acid and coenzyme A. Remember that CoA, coenzyme A, comes from vitamin B5. So, carnitine palmitoyl transferase will actually catalyze the addition of a carnitine molecule to the fatty acid, but at the same time, that coenzyme A is displaced. So you end up with fatty acyl carnitine and released CoA. Fatty acyl carnitine will then be translocated into the mitochondrial membrane, inner mitochondrial membrane, and into the mitochondrial matrix through translocase activity of carnitine palmitoyl transferase 2. So here, the fatty acyl carnitine enters the mitochondrial matrix, and together with the enzyme carnitine palmitoyl transferase 2, the carnitine is released and the coenzyme A is reattached. And so, through all of those steps, you started with fatty acyl coenzyme A in the cytosol, and you end up with fatty acyl coenzyme A in the mitochondrial matrix. You have effectively transferred the fatty acid. So that's quite confusing, that's quite long. So let's summarize. Fatty acid to fatty acyl coenzyme A activation. Fatty acyl coenzyme A activated fatty acid will then enter the outer mitochondrial membrane. Carnitine, fatty acyl coenzyme A, through the action of CPT1, there will be single substitution forming fatty acyl carnitine, releasing coenzyme A. Fatty acyl carnitine in the intermembrane space taken up by CPT2, carnitine palmitoyl transferase 2, causing fatty acyl carnitine inside the mitochondrial matrix, single substitution again, releasing the carnitine, reforming fatty acyl coenzyme A. So as you have observed, this is some sort of a shuttle. There are molecules that carry the fatty acid through the different membranes, and you end up with fatty acyl coenzyme A inside the mitochondrial matrix. Next, let's now actually go to the steps of beta oxidation. Beta oxidation simply is the degradation of the activated fatty acyl coenzyme A. Remember, this occurs in the mitochondrial matrix. So you have a sequence of steps: oxidation, hydration, oxidation again, and then thiolysis. There are 4 enzymes that catalyze beta oxidation, but all of them will be called collectively as fatty acid oxidase because the end function would be to oxidize the fatty acid. So, as we discuss the different enzymes, I want you now to focus also your attention to the right side image. We start with the activated fatty acid. That's your fatty acyl coenzyme A. Carbon number 1 is the carbonyl carbon. That's on the right, C double bond O. Going to the left, you have alpha carbon and then beta carbon. So beta oxidation is actually the oxidation of the beta carbon. And the bond is between the beta and the alpha carbon. So that's the bond that you oxidize. So we start with oxidation. The first enzyme is fatty acyl coenzyme A dehydrogenase. You remove hydrogens between the alpha and the beta carbon. So you end up with a double bond. And the hydrogen that you removed, you transfer to FAD, forming reduced FAD. The next step would be to hydrate that double bond. Okay, so we add a molecule of water, HOH, and you end up with a hydroxyl group at carbon number 3, or the beta carbon. Next, you oxidize again that bond and you oxidize the hydroxyl group. So if you oxidize an alcohol, remember, you either form an aldehyde or a ketone. But because this is a secondary alcohol, you form a ketone. And so carbon number 3, from an alcohol functional group, you'll be forming a ketone functional group with your carbonyl carbon, carbon number 3. So the third step is again an oxidation reaction, and the enzyme is 3-hydroxyacyl coenzyme A, because your hydroxyl group is in position number 3. The hydrogen that you remove from the hydroxyl group, you transfer to NAD, forming reduced NAD. And the last step would be to just cut off the bond between the beta carbon and the alpha carbon, but at the same time, you will be producing acetyl coenzyme A. That step is thiolysis, and the enzyme is thiolase. So again, what are the reactions? Oxidation, hydration, oxidation, and thiolysis. Fatty acyl coenzyme A dehydrogenases, enzyme 1 and enzyme 3 are chain length specific, which means they will only catalyze beta oxidation up to a particular length of a fatty acid. Each cycle of beta oxidation, the 4 steps, will produce a molecule of FAD, NAD, and acetyl coenzyme A. So, summary, for a molecule of palmitic acid, which is 16 carbons long, you will be able to form 7 molecules of NAD, 7 molecules of FAD, and 8 molecules of acetyl coenzyme A. And you have to pay off 2 ATP molecules during activation, so that's -2. You'll produce a net ATP yield of 106. Now, what about fatty acids that are odd numbered? Remember, each step of beta oxidation will remove 2 carbons in the form of acetyl coenzyme A. So that's simple for an even numbered fatty acid. What about if you have an odd numbered fatty acid? Oxidation of a fatty acid with odd numbers will yield, at the last step of the reaction, acetyl coenzyme A and the molecule of propionyl coenzyme A. So the body has a special way of catering that molecule of propionyl coenzyme A, which has 3 carbons. First, it will be converted to succinyl coenzyme A, which again, if you will recall, is a member or an intermediate in Krebs cycle. And that conversion requires vitamin B12. Okay? So, then, propionyl coenzyme A enters the Krebs cycle and is then oxidized with the Krebs cycle. So you have here sequential enzymes, propionyl coenzyme A carboxylase, to add molecule of carbon dioxide, and that enzyme requires biotin or vitamin B7. So, after carboxylation of propionyl coenzyme A, you form methylmalonyl coenzyme A. Methylmalonyl coenzyme A will then be reorganized or isomerized to form succinyl coenzyme A, which then enters the Krebs cycle. So if you did not follow that step, let's look at the image at the top. You have the last step of beta oxidation of an odd-chained fatty acid. You end up with the molecule of acetyl coenzyme A and the molecule of propionyl coenzyme A. Propionyl coenzyme A, you add a molecule of carboxyl or carbon dioxide, and you end up with methylmalonyl coenzyme A. You then spontaneously mutate or form an isomer from methylmalonyl coenzyme A, and you end up with succinyl coenzyme A, which then enters the Krebs cycle. In partnership with beta oxidation, in states where you have a prolonged fast or there is just excessive oxidation of fatty acids, you form ketone bodies. And the formation of ketone bodies is called ketogenesis. Ketogenesis synthesizes ketone bodies, which is an alternative fuel for peripheral tissues. Remember that in our discussion of lipolysis, muscle and liver cells can easily utilize fatty acids. But if the organism or the human is not able to eat for a prolonged period of time, and the levels of glucose or sugar in the blood goes down, there are other tissues that have a hard time utilizing fatty acids. And for those tissues, the liver will perform ketogenesis to form ketone bodies. Where does it occur? In liver mitochondria. What is our starting substance? Acetyl coenzyme A. And from acetyl coenzyme A, we produce the ketone bodies, acetoacetate, beta-hydroxybutyrate, and acetone. Which step is rate limiting? Which means, what is the slowest step? It's actually the formation of HMG-CoA from acetoacetyl-CoA 4 carbons long and acetyl-CoA 2 carbons long. So the enzyme is HMG-CoA synthase. That's the slowest step. So for ketogenesis, we have this reaction sequence. We start with 2 molecules of acetyl coenzyme A at the upper right corner of the image. We remove 1 molecule of CoA from 1 molecule of acetyl CoA, and we reform acetoacetyl coenzyme A, which is now 4 carbons long. Basically, we are fusing 2 molecules of acetyl coenzyme A. We then add another molecule of acetyl coenzyme A, and we have the product HMG-CoA. HMG stands for 3-hydroxy 3-methyl-glutaryl coenzyme A. So the 3-hydroxy substituent and the 3-methyl substituent in the molecule of HMG-CoA is found here in the middle of the molecule, as you will observe, where there is both hydroxyl group and the methyl group. HMG-CoA will then be catalyzed by the enzyme HMG-CoA lyase. So you remove a molecule of acetyl coenzyme A, yung terminal portion, and you form a molecule of acetoacetate, which is now your ketone body. If the acetoacetate spontaneously undergoes decarboxylation, because you have an unstable molecule at the right portion, you will form acetone. So you release carbon dioxide from acetoacetate and you form acetone. Or that acetoacetate, because it's unstable, can undergo reduction. It will accept a hydrogen atom to form 3-hydroxybutyrate. That's another form of ketone body, which is the more predominant form, 3-hydroxybutyrate. We use ketone bodies in the peripheral tissues. But remember, it's the liver that performs ketogenesis. But at the same time, it cannot use its formed ketone bodies because it does not have the enzyme thiopherase. So ketone bodies are oxidized primarily in the mitochondria of extrahepatic tissues, muscle, kidney, intestines, and the brain. The liver is not able to use ketone bodies because, again, it does not have the enzyme thiopherase or succinyl-CoA acetoacetate-CoA transferase. So it does not have that enzyme, therefore it cannot use its ketone bodies that it has formed. Okay, so we start with this image at the top, the liver on the left. We form acetyl coenzyme A from degradation of amino acids, from glycolysis, and from beta-oxidation. We fuse two molecules of acetyl coenzyme A and we form acetoacetyl coenzyme A. We add another molecule of acetyl coenzyme A with the liberation of a molecule of CoA, and we form HMG-CoA, or 3-hydroxy-3-methylglutaryl-CoA. We remove the acetyl coenzyme A and we form a molecule of acetoacetate. That acetoacetate can leave the liver, enter the blood, and go into the peripheral tissues. In the blood, acetoacetate, because it's unstable, can spontaneously release carbon dioxide to form acetone. And that acetone, because it's soluble in our gases, it can leave the lungs and be exhaled. Acetoacetate formed in the liver, let's go back to the liver, can be reduced where it will accept a molecule of hydrogen to form 3-hydroxybutyrate, which is the reduced form of acetoacetate. 3-hydroxybutyrate, again, is a ketone body. So it can leave the liver, go into the plasma, be circulated, and be taken up by the peripheral cells. Acetoacetate in the blood, released by the liver, taken up by the extrahepatic tissue, for example, in this picture, the muscle, can combine with succinyl coenzyme A through the enzyme thiopherase to form acetoacetyl coenzyme A and succinate. So I want you now to understand that the activated molecule here is succinyl coenzyme A. It will transfer its coenzyme A into acetoacetate to form acetoacetyl coenzyme A. And because it lost its coenzyme A, it will now undergo thiolysis so that you have activation of acetoacetate. But because it lost its coenzyme A, it will now become succinate, which again is a step in the Krebs cycle. So acetoacetyl coenzyme A now is activated and will just spontaneously decompose to form acetyl coenzyme A. So what happened here? You started with fatty acids in the liver. You formed acetyl coenzyme A's. And the ultimate goal of ketogenesis would just be to transfer those acetyl coenzyme A's in the liver back into the peripheral tissue. So you have some sort of transfer of acetyl coenzyme A's. And therefore, it's a way by which the liver shares the energy with the rest of the body. So what about hydroxybutyrate? Hydroxybutyrate, a ketone body in the blood, enters the peripheral tissues where it is reoxidized to form acetoacetate. And acetoacetate, again, just for memory, will take up coenzyme A from succinyl-CoA, form acetoacetyl-CoA, spontaneously decompose into two molecules of acetyl-CoA, where acetyl-CoA is taken up by the Krebs cycle. So, through this lesson, we have understood how we oxidize fats, and how we form ketone bodies. Thank you for listening.